Welcome this afternoon of the 6th of November for our next webinar of the Health Firm project. We are now going to send some information about microbiology, technology of sourdough production. Professor Christophe Courtin, Luc de Vuist, and Professor Katina and Valerie Gutzal will speak about the sourdough technologies. And the program is that Professor Christophe Courtin will provide a short introduction to the Health Firm project, about 5 minutes, let's say, and Professor Luc de Vuist will give more information about microbial aspects of sourdough production, and Professor Katina goes more into the details of technological and sensory aspects of sourdough production, whereas Dr. Valerie Gutzal is providing you information about applying this knowledge into fermented foods. I do have some short household rules. That is that presentations will follow without any interruption. You as a participant can post your question in the question box. There is a question box in the menu. And if you have a very specific question to one of the presenters, please notify the presenter's name, then we can address the question to the specified presenter. And at the end of the webinar, all the questions will be collected, and we try to address as many as possible in the question and answer session. However, if there are some questions that are not answered during the Q&A session, we will try to follow up on these in a written way. With this having said, I would like to invite Professor Christophe Courtin to give his short introduction to the Health Firm project. So please, could you make Professor Courtin presenter? Thank you.
Thank you, Jan. Can you see my slide?
Yes.
Yes, perfect. So very good afternoon, everybody, and thanks for joining us for this webinar from Health Firm. I'm indeed Christophe Courtin, so project coordinator of the project, of the Health Firm project, and it's a pleasure to introduce to you, or to those of you who are not familiar with the project yet, the project in a few slides. So we are dealing with a European project, Horizon Europe. We have 23 partners on the project and a budget that's a little bit over 13 million euros and a project duration of 4 years. You can see all the different partners in the lower left-hand corner of your slide. I'm not going to go over all of them, but I think you can acknowledge that we have quite a nice group of partners gathered for this project. Now, what is the project about? On the lower part of the slide, you can see the main aim of the project, which is to enable an evidence-based transition in society from traditional and sustainable plant-based fermented foods and drinks that deliver health benefits to consumers by design. Now, this is a mouthful, of course, and in bold you can see the different elements that are the most important. So it's evidence-based. We want to do research to support the transition that we're suggesting from traditional to sustainable plant-based fermented foods. And there's a large focus on health benefits and on trying to do fermentations by design. So not relying on the traditional spontaneous fermentations, but going to build consortia. And for this, of course, we need microbial resources, we need fermentation technology, we need to investigate human health and microbiome, the link between those, and we also need a lot of knowledge on consumer behavior. And that translates into a scheme that you very often see for this type of European projects, where everything is gathered in one comprehensive slide. So again, on the top, you see microbial resources coming back. I'll come back to that in a second. We have fermentation technology, where we go from raw materials to fermented foods and fermented ingredients. We have a section on human health and microbiome, where we are proud to have 5 full-blown intervention trials at 3 locations in Europe, linking whatever happens in those trials, also with the gut microbiota and also with in vitro trials, to try to understand what is happening in vivo. And again, we have also made sure that consumer science is interlinked with each of the work packages that I just mentioned before. And then, of course, we have two flanking elements, which is project management and dissemination of results. And that is something that we are doing today. We try to share knowledge that we have on specific topics with you as a public. Two things. Going to pilot scale is important. Why? Because we have intervention trials, because also the aspect of upscaling is important. And we also put a lot of efforts in omics technologies. I would like to finish this very short presentation of the Health Firm project by looking at a small part that is very relevant for what we are having today as a webinar. And that is the community science project that we have built into the Health Firm project. Now, that community science project is centered around sourdoughs, and we have set out to collect sourdough samples from all over Europe. And we have recently closed the project, or the gathering part of the project, and we have managed to get 680 sourdough samples and more from all over Europe. So we're quite happy with that. And at this moment, a lot of the research groups are busy on disentangling those sourdoughs or the microbiota in them, identifying microorganisms. And the idea is that based on this, we will come up with novel strains that have desired functionality in grain-based fermentations. This is not the only way of getting new strains, of course, but this is one of the important elements. And so this community science approach is mostly based on sourdough. And with that, Jan, I would like to give the floor back to you to introduce the next speakers. And I wish all attendees a very enjoyable 1.5 hour learning about sourdough microbiology, technology, and application.
Thank you very much, Christophe. Thanks for this short but very nice introduction to the project. I hope that participants understand what we are working on. With that having said, I would like to give Professor Luc de Vuist the floor. He's a professor at the Vrije Universiteit of Brussels at the Department of Bioengineering and Sciences, and already for years a specialist in microbiology. Luc, the floor is yours. You're muted, Luc.
I don't. Do you hear me now?
Yes, I hear you now.
Okay. And do you see the screen?
I see the screen. Yeah.
Okay. Thank you, Mr. Chairman. Welcome to a presentation on the microbial aspects of sourdough production. I will start with the basics. What is sourdough? I will give some examples of stable microbial consortia, and if time still allows, I will also focus a little on the health promoting potential. But let's start. What is a sourdough? Many of you maybe have made sourdough yourself. You need water, you need flour, you just mix it in a 50-50 ratio, and you let ferment it, for instance, at room temperature to have a sourdough. So it's a live, flour fermented microbial ecosystem. Now, why do we do that? Of course, to make baked goods such as bread, but the sourdough will have a contribution to the acidity, the aroma, the texture, the shelf life, and maybe the health promoting properties. We produce it at household level, but that happens under less strict conditions for fermentation and maintenance. When we do it more industrially, in big companies or at bakery level, there is a much more tight and efficient control. And indeed, your sourdough can be rather firm, or it can be rather liquid. That's why we make a distinction between different types of sourdoughs. The first one is just your water flour mixture that you let ferment, and you're going to backslop it. Every day you refresh it. And this refreshment, if you carry it out about 10 to 12 days, you will have a stable sourdough. But where do the microorganisms come from? They come from the floor, other ingredients, the environment, etc. So it's a spontaneous growth and gathering of the microbiota. While in a type 2 sourdoughs, which is more produced at industrial level, we're going to inoculate the water-floor mixture with a starter culture of mostly lactic acid bacteria. So the microbiota is driven by the performance of that starter culture. That starter culture will finally give you the end product. Here you do not apply backslopping. This is a long fermentation process. It can last until 72 hours and you get the sourdough. And in a type 3 sourdough production system, you're going exactly to do the same, but the sourdough obtained, you're going to backslop. So the microbiota that will dominate the sourdough during the backslopping days will depend on the competitiveness and the robustness of the microorganisms that you have inoculated by means of the starter culture. So type 1 sourdoughs is mostly at bakery level, type 2 sourdoughs at industrial level, and type 3 sourdoughs can be at both levels. So the baked goods containing sourdough, of course, are breads, but it can be also baguettes, cakes, biscuits, snacks, pizzas, etc. But concerning bread, I would like to make a distinction between what I call sourdough bread and bread with sourdough. In a sourdough bread, the bread is made from the sourdough. So it is not bakery yeast, but the sourdough itself that enhances the volume, that makes the texture, that has the leavening capacity. So there is no baker's yeast added. Of course, to have some volume, we sometimes allow the addition of bakery yeast, but maximally 0.2% on a mass for mass on floor basis. A bread with sourdough, on the other hand, is where the sourdough is used as an ingredient. 10%, 20% of the mix can be sourdough. And there we are going to add the classical common baker's yeast to have leavening. So the live sourdoughs contain active microorganisms, whereas dried sourdoughs that also can be obtained are to be considered as inactive sourdoughs. So as I just said, it's a microbial ecosystem, and the microbes that occur are lactic acid bacteria and yeasts. And they usually occur in a 100-to-1 ratio. So there are more lactic acid bacteria and yeasts. And which lactic acid bacteria do we have? Well, if we do an investigation of hundreds of wheat and rye sourdoughs, the most occurring ones are those Fructilactobacillus sanfranciscensis, Campanilactobacillus parliamentarius, etc. Whereas for the yeasts, it mainly is Saccharomyces, yes, the same name as the baker's yeast, but it can be naturally present as well. Maldiosema, that the former name was Kazakhstania, Vicergamomyces, and Pichia. These are the most important ones. If we perform a statistical analysis on that, we will see that the variation in type 1 sourdoughs is much higher, but still determined by the occurrence of that most frequently participating lactic acid bacteria species, namely Fructilactobacillus sanfranciscensis. If we do the statistical analysis without and with Fructilactobacillus sanfranciscensis, you see that the variation is much bigger without than with, because it's the most occurring lactic acid bacteria species. It is, however, and we wanted to have that, not a region-dependent ecosystem. So the geography or the region doesn't determine. We believe that San Francisco bread is only made in the Bay of San Francisco, that certain Italian sourdoughs or Belgian sourdoughs have a specific microbial ecosystem. We have investigated that, and that's not the case. That's why in the Health Firm project, we enlarged that to the whole of Europe to see if there could be a link. But the decisive impact is the raw materials, the process parameters applied, the dough yields, the oxygen tension, mixing or not mixing, small or large bubbles, etc., etc. So many process parameters are actually determining the ecosystem and not the region itself. However, if we do sourdough productions in a bakery environment or in the laboratory, we do see a difference. In the bakery environment, we mainly have Saccharomyces cerevisiae, and that's because in that bakery also yeast-leavened breads are produced with baker's yeast. While in the laboratory, we hardly see Saccharomyces cerevisiae, but we rather observe Vicerhamomyces anomalus that is present in the flow. So there could be a difference because of the house microbiota that is present in the environment. Third, we have, thanks to the growth of the lactic acid bacteria and acidification, lactic acid bacteria can produce either solely lactic acid, we call it homolactate fermentation, or a mixture like lactic acid, acetic acid, mannitol, etc., which is called heterolactic fermentation. While the yeasts also produce some organic acids, in particular acetic acid and succinic acid, and that you can see here in the pathways. So for lactic acid bacteria in particular, so the most of lactic acid bacteria are maltose negative, and the yeast is maltose positive. But in a sourdough, it's the opposite. It's often the opposite. The lactic acid bacterium is maltose positive because it has a very specific enzyme which we call maltose phosphorylase. And through that pathway, finally ethanol or acetic acid and lactic acid are produced. Well, if there is an additional ingredient, for instance, citrate, they can do a co-fermentation. So together with the maltose, they're going to ferment the citrate, and then we end up, of course, first in pyruvate, but then in different other compounds which have more buttery notes. So the lactate is producing a fresh sourness, like in yogurt, the acetate a sharp sourness, and diacetyl and acetoin, which are the result of that co-fermentation. So with additional pyruvate, you have buttery notes in the sourdough. The acidification is also responsible for the fact that the gluten proteins will have a higher solubility. And due to the acidification, the endogenous enzymes present in the floor will also do their work, because the lactic acid bacteria are not really protease positive. They have peptidases, but not extracellular proteases. So we have, thanks to the acidification, an interaction between the microorganisms and the environment. And indeed, a disadvantage could be that the gluten are depolymerized, enhancing their solubility, enhancing their susceptibility for proteolysis, but making it more difficult to produce a firm dough. And that's why process parameters such as fermentation temperature, fermentation time, dough hydration, and gas retention will play a very important role. There are, of course, also oligosaccharides naturally present, either produced by the lactic acid bacteria, like glucans, fructans, and other oligosaccharides, or naturally present in the flour, like arabinoxylans. Of course, more in rye flour than in wheat flour, but they will also help to have a network that allows to make an interesting dough. Finally, the lactic acid bacteria, of course, will contribute to flavor. I already spoke about sourness and buttery notes, but they can also do a lot of amino acid conversions, making other notes like malty and fruity tastes. And the yeasts, of course, can do that in particular. They will, of course, also make some organic acids, but they will mainly convert these amino acids into malty fruity notes and further into esters. That's illustrated here. So lactic acid bacteria can convert the amino acids through a series of reactions that results in aldehydes and alcohols, and these give these malty and low fruity notes. But the yeast can go further. They can, after these conversions, they can condensate the alcohols with either acetate to produce acetate esters or the ethanol to produce ethyl esters. And it is these ethyl esters and acetate esters responsible for the fruity flavor of sourdoughs. Of course, that should end up in the bread after the fermentation. The bread is baked. and then the volatile flavors can disappear. I don't have the time to go into details, but optimizing the conditions can avoid that. But an interesting thing to mention is the ADI pathway, the arginine deaminase pathway, which is present in lactic acid bacteria occurring in sourdough as well. They can take up arginine, convert it through citrulline and carbamoyl phosphate into ornithine. Also ammonia and carbon dioxide are produced, but this ornithine is again secreted. And when this can react when decarbonyl compounds are present during the baking process, taking place at, for instance, 200 degrees, they can make other compounds. And one of the most interesting ones is 2-acetyl-1-pyrrolene. That's the typical crust aroma. That is what you also smell when you enter a bakery, that typical compound that characterizes a bakery environment. Health promotion is of course, sorry, I forgot to say, except for flavor, these lactic acid bacteria also help in texture formation, and for instance, exopolysaccharide production. Some of these amino acid derivatives have antibacterial and antifungal activities. Of course, the yeasts provide a good texture to the bread, and also their antifungal activities can be present. So then is the question, which microorganisms are present in the sourdough? Well, we have seen through the years by studying different sourdoughs that some lactic acid bacteria are very well adapted. I have given you already the example of Fructilactobacillus sanfranciscensis as the most occurring one, because indeed it has an efficient maltose heterofermentation, an adapted amino acid metabolism, and it can even withstand different stresses that are occurring in the sourdough matrix. The same actually is the case for Limosilactobacillus fermentum. It also does maltose heterofermentation. It has this extra arginine deaminase pathway, and it can even produce some oligosaccharides that contribute to the texture. Of course, there is also a yeast present, and what do we see? Most of the time, a certain lactic acid bacterium is associated with a certain yeast. In this case, Fructilactobacillus sanfranciscensis is associated with Madiosima humilis or Madiosima exigua. And why is that? Well, as I told you, normally the yeast is maltose positive and the lactic acid bacterium maltose negative, and here it's the opposite. Thanks to the maltose phosphorylase activity, the lactic acid bacterium is maltose positive and the yeast is maltose negative. So they can occur, can grow together, can perform their activities together. Indeed, the yeast uses the glucose while the Fructilactobacillus uses the maltose. There is also fructose present, and this fructose can serve as an alternative acceptor for the lactic acid bacterium, making mannitol. Where is this fructose coming from? Thanks to the activity, the invertase activity, the beta-fructofuranositase activity of the yeasts that can elaborate fructose. Also, amino acids and vitamins are elaborated by the yeast cells, and yeast cells can lyse during the process, and all these nutrients are then available for the lactic acid bacterium. The acidification causes, of course, acidic stress. Production of acetate can stress the yeast. Normally, a yeast will start to sporulate, but this is not the case because there are acetic acid tolerance mechanisms present in that yeast, so that no sporulation needs to occur. And the same, the yeast can cause oxidative stress by thiol oxidation, but thanks to an oxidative stress tolerance by the lactic acid bacteria, making the redox balance optimal for the growth of that bacterium, this can tolerate this oxidative stress. So these microorganisms can nicely grow together. And this is not the only consortium. There are other Madiosima species like Barnetti that also make this association. There are even associations between Lactiplantibacillus plantarum and Saccharomyces cerevisiae. There are some lactic acid bacteria that often occur together. The name says it here, Compagnie Lactobacillus parliamentarius, companion, so it is occurring together with another species. And there are even sourdoughs known with a very complex lactic acid bacteria yeast consortium, as the last example here mentioned. We developed during a project also a new combination of Compagnie Lactobacillus crustorum and Vicerhamomyces anomalus. So our yeast is maltose positive, but it is especially selected because of the production of fruity esters, and it is also acid tolerant. While the Compagnie Lactobacillus crustorum is homofermentative, produces mainly lactic acid, but does have this maltose phosphorylase, even produces buttery notes, and of course will cause the acidic stress to which the yeast is acid tolerant. And as you can see here, we can produce nice breads with that, and tasty breads, of course, with that consortium. I still have 2-3 minutes, so I can very short mention the health potential. We know that sourdoughs are promoted as being health promoting. Why is that? Because they elaborate amino acids. As you can see here, in a sourdough you have much more free amino acids than in a non-sourdough environment. They can produce special amino acids like gamma-aminobutyric acid that have a bioactivity in the human body. Other bioactive peptides can be elaborated, phenolic compounds, vitamins, minerals, etc. I have given here the example, but there's one thing that you should take into consideration. We do not eat sourdough, we eat breads. And of course, from sourdough, you still make the dough to make your final bread and bake the bread. So the question is, what happens with these amino acids finally in the bread? So that's research that still has to be done further in order to make some nice conclusions. Another example is phytase activity. So there is phytic acid with bound phosphate. This can be elaborated. This can be contains minerals like iron, calcium, magnesium, manganese, etc. that are not bioavailable. Thanks to phytase activity, this is broken down and these minerals become available. So there are more minerals present in sourdoughs. But the question is, what is this phytase activity? Where is that coming from? Is it a microbial phytase or is it the endogenous phytase of the flow? We could show that it is mainly the endogenous phytase that is responsible for that. Thanks to the acidification, the optimal pH will be there to have a maximal phytase activity. And the last example, there is sometimes we read there are more phenolic compounds present, but please, that's not correct. Lactic acid bacteria do not produce phenolic compounds, but what they can do is polyphenols that are present, they can modify them because they have esterase activity, they have reductase activity, they have vinylphenol reductase activity, and that way you can, of course, larger polyphenols modify into shorter ones, and that give the apparent increase of the phenolic compounds, which then can have that antioxidant potential. What we are still missing is convincing human intervention trials and clinical studies. I prefer human intervention trials with healthy individuals. There are a few. Here is a list of those that you can find in the literature, that there are many more, but that are rather done correctly. But if you look into the results very carefully, I have to disappoint you and say, yes, there are conflicting evidences. Only in this one, a clinical study with individuals with IBS, there was some evidence that sourdough bread would be more health promoting than normal bread. But of course, we are still continuing with that research to show if there is an increased health promotion by consuming sourdough bread in comparison with yeast bread. I would like to thank many people who have collaborated to our sourdough research of the last 30 years. Many PhD students have defended their PhD on that topic. These are still busy. Those have done that in the past. We have a lot of collaborations with other universities working on taxonomy and on dough technology, etc. And of course, many financial supporters, among which the Health Firm project, co-funded by the European Union. I thank you for your attention.
Thank you very much, Luc, for this excellent presentation. I understand that there is a numerous possibility to develop nice and potentially healthy sourdough products. I hope that the people that are listening are also posing some questions. There was an option to pose your question in the question box. Thanks again, Luc. I would like to give the floor to Professor Kati Katina. She's a professor at the University of Helsinki at the Department of Food and Nutrition, and she has a tremendous background in sourdough breads. Kati, floor is yours.
Thank you. Can you hear me?
Yes, I can hear you. Yeah, thank you.
Okay, good afternoon, everybody. So my topic is to introduce you technological and sensory aspects on sourdough production. And a little bit about my background. I have worked with the fermentation of different types of grains, and especially sourdough fermentation, over 25 years. And my studies have dealt with fermentation of wheat, rye, oats, and lately faba bean. So if you think sourdough process as such, you can even try to do this at home, and it's quite popular actually to do it home. Taking into account in the Professor Luc de Boost's nice presentation, there is very interesting microbes, bacillus, bacteria, and yeast, which are needed. And when you combine them with the different types of grain flours or grain fractions, you can use white wheat flour, you can use wholemeal flour, you can use white wheat flour mixed with different types of alternative grains, you can use bran fractions, and you end up after fermentation in different after fermentation temperatures and times, you end up with a sourdough. And what happens actually in a sourdough from grain perspective is rather interesting. Of course, there is this altered microbial community, which Luc revealed already. And then there are a number of changes what happens actually in the grain-based components. Typical changes can be actually changes in polysaccharides, both non-starch and starch polysaccharides. For example, arabinoxylan and beta-glucan are often changed during fermentation, the main fiber components. Phytochemicals are changed, they might be liberated from grain-based raw materials, especially in the whole grains and from bran layers. And what definitely happens is that in a sourdough system, enzyme activities are altered. They can be from microbes themselves, or they can be sitting in your raw material. And sourdough system is either activating or inactivating them. Then thinking about sourdough wheat bread or sourdough bread in general, you can introduce your sourdough at the level up to 40% to your bread. And if all these changes, what you can induce with the fermentation are at, you could say, at optimal position, you could have an improved volume, safety, shelf life, nice mouthfeel, nice flavor, and even increased bioactivity in your bread. But if you look a little bit more closely, what is actually behind the texture and flavor? All grain-based raw materials, whether it's a bran layer, whether it's a whole grain flour, whether it's a faba bean, have all very different compositional features. So there are different chemical compounds sitting in a raw material. Then there are polymers, which have different properties. There are tissues and structures and interactions of different tissues and structures. And all these raw materials have typically different type of particle size and even particle shapes. which sort of makes them different, how they absorb water and what kind of mechanical properties they have. And interestingly, if you think flavor and texture forming properties, all of these components in a grain matrix are influencing the sensory quality and texture of the food. And these same features are also heavily connected to physiological functionality, especially as a dietary fiber component, which sort of are influencing the health outcome of grain materials we eat. And fermentation, microbes and enzymes sitting in a fermentation system can alter actually all of these. And often these changes happen at the same time. So we are changing chemical composition and polymeric properties, which have at the same time, they are influencing both sensory quality and texture forming properties and also nutritional properties. And they are actually rather hard to separate from each other. But sourdough system and fermentation in general is a multifunctional system, and typically it changes many things at the same time. So a little bit closer look about fermentation-induced changes, especially related to flavor and raw materials. But in sourdough system, as I said, there are always endogenous and indigenous enzymes, either from raw material or from microbes, and other microbial activity, metabolic activities, which Professor Wust actually showed already. And typically there are formation of acids and pH is reduced in lactic acid bacteria fermentations, and that is influencing the enzyme activities. So typical changes are proteolysis, so proteins are solubilized, partly degraded. This is of course depending on the starter, strains sitting there, fermentation conditions, and other processing parameters. So proteins are typically degraded, at least in some extent, and also cell wall are hydrolyzed, especially when we have fiber-rich raw materials to be fermented. And also there are changes in starch. So all major structure forming components are modified during sourdough fermentation. Other interesting feature is formation of metabolites. Especially interesting is flavor active compounds which are created. And these are really the strain specific and fermentation condition specific, that there can be hundreds of different flavor active compounds which are created during sourdough fermentation and fermentation in general. Also, other metabolites can be created. One type, what I have been involved studying, is exopolysaccharides. So these are polysaccharides which microbe is able to produce during fermentation if the conditions and substrates are correct. And now I will give you two examples how the sourdough can work in influencing bread texture, and my other example is flavor of a bread. And I have been studying especially wheat bran and whole grain flours because they have a very high nutritional quality. And they are interesting only not because of fiber, because there are not only fiber, there are also phytochemicals like phenolic compounds, there are minerals, there are vitamins, which usage of whole grain flour and getting enough actually cereal fiber is highly recommended by nutrition experts. And related to this study, what was done for some time ago, we studied possibility what happens if we take the bran fraction, outer layers of the kernel, and ferment with that and introduce it back to the wheat baking. And that study was done with the two microbes, Cassastania exigua and Lactobacillus brevis. And there was a shorter fermentation and longer fermentation with or without hydrolytic enzymes. And then these fermented so-called bran sourdoughs were introduced to wheat bread and compared to the same bread where there was untreated native bran. And then the bread was analyzed, where specific volume, texture, and shelf life was analyzed. And what did we see? Typically, here on the left side, you have wheat bread, no bran, which is a control, and it's a very nice fluffy wheat bread. And then you add 15% of bran, the volume goes down. So bread is really dense, as seen in this picture. And then sourdough fermentation, when it works optimally, it can increase the volume back to the level where actually the wheat bread was, without addition of bran. So either without or with enzymes, we can improve the bread volume. So significant increase in volume. And also related to the texture, you have there is a bread in a baking day, 0 days, and bread after 4 days of storage in room temperature. You see that in day 0, the wheat bread is really soft and it's getting much harder, and it stales. Bread is getting harder and dry, and other staling related phenomena happen. And typically we follow it by measuring the hardness. If you introduce untreated, the native bran, the changes are even more enhanced. So bread is even harder after the storage, and already in a baking day, bread is harder. And the same sourdoughs, which improve the volume, you can see that depending on the type of sourdough and addition of enzymes, you actually can create softer bread texture and especially retard the staling, so hardening of bread, when you use the sourdough technology. And why this happens? One of the expected reasons is that fiber components in high fiber raw material are altered. And this is example of bran fermentation, rye bran fermentation in this study. But in native rye bran, you have relatively little soluble arabinosilans. They are mainly insoluble. And after the fermentation, with enzymes in this case, you have significant increase in amount of soluble arabinosilans. So this is actually what happens in sourdough. When you introduce this fermented bioprocessed bran to bread, and of course there is not much, if you use native bran, there is not much soluble arabinosilans. But if you use the fermentation technology, you increase, of course, the amount of soluble arabinosilans in bread. And how this is actually seen in structure of a bran and bread. Here you have a protocol where you combine enzymatic treatment with fermentation, and you see here on the upper corner on the right, you see intact bran cell wall structures. This is actually aileron layer, but this blue colored walls are actually containing dietary fiber and arabinosilans and beta-glucan, and red one is protein. And so the protein and other components are locked inside the cell wall structures. And when you do the fermentation, you can notice that these cell walls start to be degraded. So we are degrading and modifying the fiber component part. And the same thing is seen in bread, that you actually create quite a lot of changes in grain cell wall components during the sourdough fermentation. So sourdough technology can influence positively both the texture of the bread and volume of bread, and even retard the staling, which is of course very good if you improve the shelf life of bread and you don't create waste which should be discarded. So my second example is influence of sourdough on bread flavor. And sourdough is typically used in white wheat baking to create nice flavors. And that is sort of one of the original reasons to use sourdough, except that it improves also shelf life in terms of microbiological safety. But what I wanted to present here is case of faba bean enriched bread, because all of us know that we should eat more plant-based proteins like soya, faba beans, and peas. But unfortunately, they have some flavor challenges, and not all consumers are big fans of these beanie flavors, which are related to oxidation of fatty acids, and especially the bitter taste and astringency, which is related to tannins, phenolics, saponins, free fatty acids, and fatty oxidation products, are quite hard to tackle by consumers. And also there is a very strong aftertaste if you use significant amounts of soya, faba beans, or peas, for example, in bread baking. And maybe one of the most challenging flavor issues is bitterness, because flavor chemistry is grain specific behind the bitterness, and it's rather complex. So it's not only one compound creating the bitterness, there are several. There are a lot of individual differences, so sensation or ability to sense bitterness is different from one person to another. And originally, in the history of mankind, bitterness is a warning signal that do not eat that this might be harmful to you. So these kind of taste challenges have to be addressed if you, for example, would like to make a high protein bread, but it cannot taste bitter, or there cannot be unwanted tastes if you really want to consumers to use this type of bread. And of course, it's a cultural thing. What do we like? White bread is very, very popular in a European context and in a Western diet context. And whatever you add there, alternative grains or grains which consumers are not familiar, you change both texture and flavor, and very easily end up with unpleasant taste and unacceptable product. And as good as sourdough technology is, it's typically based on acidity formation. And these hydrolytic reactions, which I already mentioned, like proteolysis, degradation of cell walls, they may actually lead to release of peptides and phenolic acids, which makes actually bread, for example, even more bitter and an even stronger aftertaste. So not all fermentation improve a flavor immediately, but it depends on what kind of grains you are fermenting, and what are your fermentation conditions, and what kind of strains you are using, what kind of consortia you are using to do the fermentation. So it's a interaction of all of these factors influencing on taste. So it's really the tailored fermentation needed to create nice flavors, especially if you're using these alternative grains in bread baking. And what we have studied quite a lot is actually production of these exopolysaccharides. Exopolysaccharides can be produced by certain lactic acid bacteria, like Weisela confusa from saccharose. And these dextran exopolysaccharides, like dextran, can have both structure forming and flavor influencing impact on bread. And what do they do? Exopolysaccharides, they increase viscosity of sourdough, they impact rheological properties, they have positive impact on specific volume, crumb softness, they can retard the staling, and even can have an interaction in the flavor perception of a product. And in this particular study, we fermented fava bean flour with 4 different strains, 2 Lactobacillus plantarum strains, more typical sourdough strains, and 1 with the Pediococcus pentosaccheus, and then we had this Weisela confusa plus saccharose addition to produce dextran in that particular sourdough. And then we analyzed the bread texture and did the sensory evaluation with quantitative descriptive analysis by using the trained panel to see what happens. And volume-wise, you can see here that this is a control wheat bread, and then you introduce the fermented fava bean with, let's say, more traditional strains, even adding the native untreated fava bean, already decreased volume, and sort of traditional sourdough fermentation did not do much impact on the volume. Actually, it was a little bit even lower than in control bread with the untreated fava bean. But with Weisela confusa fermented sourdough with the dextran formed, we had a very nice volume, and comparably we have softer breads. So compared to the control bread with a native faba bean and also compared to other type of more acidic sourdoughs made with more traditional strains. And flavor-wise, you see that the black line here is wheat bread supplemented with native faba bean, and red line is Vesla Confusa sourdough, and more traditional ones are yellow, green, and gray lines. And you see already by just looking with overall sensory characteristics that these breads are quite different. But with traditional acidic sourdoughs actually increase some flavor notes like beaniness, musty odor, and sour taste and aftertaste. And if you use this dextran sourdough technology, they actually have less beany, less musty, and less intensive odor in the bread, and less aftertaste compared to other types of sourdough fermentation. Of course, we had also a better bread texture. So, in conclusion, I would like to say the take-home message is that sourdough fermentation and fermentation in general can efficiently improve volume, texture, and shelf life of bread as well as flavor. However, this is very much strain dependent. It really depends what kind of strain or strains, actually the consortia you are utilizing, how it's matching with the raw material you are using, and it's very much raw material specific. So I could say that each raw material actually requires certain defined consortia and defined fermentation conditions to obtain this optimal influence of sourdough. And I'd like to thank you for your attention. This is my group at University of Helsinki, and if you are interested in fermentation and grain-related research, please visit our website.
Thank you very much, Kati, for the very nice presentation. I understand from your explanation that there is still quite some challenge to develop new sourdough breads based on different sources. And I would like to invite Valerie Goodsall. He's a senior scientist for plant-based applications in Novonesis, and he has a background in microbiology and specialized in fermentation processes. And he will go more into detail on how to use matching plant-based cultures. Valerie, the floor is yours.
Good afternoon, everyone. I hope you can hear me and see my presentation.
We can hear you, and your presentation is not yet in presentation mode.
Give me a moment. Is it working now?
Yeah, it's working now. Thank you. Oh, it's going back. Yeah, now it's working. Yeah.
Okay, great. So Katina gave a great introduction of how it is important to consider different microorganisms and different bases that are aside of the traditional sourdough making process. And I will give you a little bit more understanding of what fermentation do and how they are designed in going from smaller scale or kitchen to a production. My agenda will include main aspects for fermentation design, base and base modification through different methods, sugars as a main carbon source for the starter culture and their uptake during the fermentation process. We also would go into details about how we go from smaller scale in designing fermentations to larger scale, going to pilot plants and further up to the production process. And of course, as we say that the taste or the flavor is the king, we do sensory evaluation. I'm not sure if I'm still sharing.
Somehow your presentation switches into the other mode.
I don't know why. Okay. Now is it working?
Now, yes,
great.
It works.
I'm continuing. So basically, when we talk about fermentations in plant bases, we should really consider one of our biggest enemies here are the Bacillus cereus, and they could heavily multiply in unsoured material, causing quite some of damage to the manufacturing process and the consumers themselves, right? So what we do is we ask to design fermentations for our customers and ourselves when we do that. So the fermentation process could reach pH of 5 within a maximum of 7 hours. So it has to be a quick fermentation, having in mind that the bacillus counts of Bacillus cereus counts, especially in the raw material, are below 100 CFUs per gram. However, in the Bacillus cereus case situation, the lower you go in the pH, the better. So heat treatment is applied. A traditional dairy material is often not enough. So when we talk about plant bases, it has to be a really harsh temperature treatments, over 92 degrees for quite a period of time of 15 minutes, 20 minutes, and longer. And then another aspect is the process design. So we need to have in mind all the other process steps that are taken to make sure that either heat transfer is correct, that the emulsification stability of the product is right, and mixing in the culture and pH probe location is right, because we want to design a good process. And the process fermentation is, as shown here, normally contains lag phase that could be either non-existing or could be really long, or the exponential phase. That's where our starter cultures grow. And the final, or cut pH, as it's called in industry, is the one where we want to stop the fermentation. Here on this graph, you see that the safety threshold of pH 5 has to be reached within 7 hours. And you can clearly see that the curve on the left, the blue one, goes perfectly fine, and the green fails the requirement. When we talk about base and the base composition, we talk about different levels of proteins. We talk about different structure of proteins. It's a different type of level of fat that has an impact. There are also fibers that make material more thick and more difficult to process and mix in during the manufacturing process. There could be stabilizers added that could impact the fermentation process. Water plays a big role too. However, one of the main impact factors besides fats and proteins that give a buffering capacity of the whole mix are the fermentable sugars. So sugars need to be accessible to the starter culture. And when we design starter cultures, normally industrially produced starter cultures for fermented plant-based products, historically, they are originating very often in dairy materials or in dairy fermentates. And when we are going through this transition process, we need to keep in mind that plant-based is so many times more difficult to understand and to analyze than the dairy material that has been in study for around 200 years now. And through some of the tricks in biochemistry, we could modify the plant base. And one of those great tools for us to design a better base that is easier to ferment are the enzymes. These enzymes, they could be added to the heat-treated base, unheat-treated base, and they can create a range of differentiated bases for from simple raw materials to very complex ones. Through enzymes, we can adjust the sweetness in a final or intermediate base product. Production could be simplified. The nutritional profile could be changed to better match the target consumer. We could adjust mouthfeel and the viscosity of the beverage, or vigor or sourdough, if that is the case. We could be improving yields and, of course, speeding up the process. So to show you very quickly of what enzymes could do in the process, I offer this 10-second video where we add amylase into a very thick base, and you can see how it modifies it. There is no audio in this video, so it's just bubbling. So here we're adding amylase. It chops down the starch in the base, and voila, from the very thick paste, we have very liquid, easy to flow base to work with further down. By the way, this process is widely used for making oat drinks. And each base is unique. Kati mentioned that several times, that we need to watch really carefully of what we are working with. And on this graph, you can easily see that the three very popular bases that are used in the industry now, oat, soy, and almonds, they are totally different in their sugar composition. And I mentioned that sugar is a key source of is a key source of carbon, and as a key factor for fast and robust fermentation process. On this table, you can see that different species or different microorganisms, they tend to like different sugars better than other sugars. However, when designing the fermentation, and if you do not know how well the species of microorganism is going to taken up any specific sugar that you have in your base, and you maybe didn't do any analysis, or you're not planning it, and you want to simplify your development process, the easiest way to go forward is simply to add glucose, because glucose is a universal tool to speed up any industrial fermentation. Also, sucrose is a very good material for that, because as you can see on this graph, for example, some of the species that we use to develop our starter cultures, they are totally fond of these two sugars. When it comes to fermentation of different bases, they are definitely not the same when going from soy to oat to almond and coconut. You see that these fermentation curves, they change a lot as we go from highly protein material like almond and soy to a lower protein material that has a lower buffering capacity, that is oat. And keep this buffering capacity and the sugar profile as key differentiators when it comes to base selection. However, even going into the same base like pea and fava, we see clear differences in the fermentation process when we compare different manufacturers of the pea protein and fava protein concentrates and isolates. Fermentation looks quite different from one to another. We get different profiles, and you see, if we talk about the safety threshold of pH 5, this threshold is reached at quite different time points, and for some of them it's longer than for the others, and for industrial fermentations that are usually ending up around 4.6. For example, these two blue lines, they are far they're taking far longer than the other ones. Talking about glucose as a universal sugar source, as a universal way to speed up fermentations, here what we see is a green fermentation curve where we added glucose in the base. Here it is soy milk with 0.02% starter culture, and the red one is the base without glucose. So when all the accessible sugar for the starter culture has been consumed, pH didn't move any further from that standpoint. And you see that after 7 hours, pH was still higher than 5.8, meaning that there's a very, very high risk that material is contaminated with, if material was contaminated with spores, that the spores would have started growing and the base became unsafe. So what actually happens when we add glucose during the fermentation process? So if here you see the fermentation curve with glucose to start on, it goes nice and smoothly down from the first minutes of the fermentation. We can still fix if something is going wrong by adding glucose in the process. And here it shows in the case of P isolate base, where there was basically no sugar that is taken by the starter culture. Then when adding glucose, fermentation starts going really fast, and the pH drops also very fast. How do we think of all these aspects when we talk about designing a starter culture? What do we need to consider? What are the species that we could use? You know that the strain banks these days, they contain thousands and thousands of different strains. And Katja was also mentioning that we need to test specific strains in a specific basis all the time to make sure that the combination is something that is usable for us, is something that gives us a great tasting product, and the fermentation that is speedy enough. So what we do in our company, we designed a combinatorial microbiology approach to test different strains in the high throughput methods. For that, we take the base and we fill it up into a microtiter plate, where we add color indicator, and then this color indicator is measured in the 96 well plate by robots and equipment that can read the color change. And based on the color change, we can relate it to the pH change in the tube or in this specific well. From this machine, we get a graph that looks like this, that includes the most important part of the fermentation process, so pH from 6.0 and below, showing us a fermentation time or the time to the target pH, the shape of the curve. We can also understand how big is the post-acidification potential. Post-acidification is something that is also very considered in the industry to understand how sour product gets during the storage process. And of course, since we're using robots to move things or move samples within the same microtiter plate, we can also assess texture by the suction pressure that is needed to suck in and suck it out of the 96 well plate. So here you see that the target pH is, for example, 4.6, and the time to target pH 4.6 is quite different from one strain to another. But how would we usually change this or share this findings with within ourselves, or how would we assess it better? So on the bottom, we usually list our cultures, and on the Y line we do the time to pH. So basically, it's this exactly time that curve needs to reach the pH 4.6 or 5 or 4.4, depending of what we're aiming for. And here on this graph, you see that, for example, this specific starter culture reaches pH 4.6 at 10 hours 45 minutes, or sorry, pH 5 at 10 hours 45 minutes. And on this kind of plot, we could compare so many different starter cultures all together, which gives us a lot of flexibility to work with. And post-acidification, we also can measure what is the pH of the sample when it reaches the target pH plus 3 hours. So different combinations of starter cultures, they give different changes in time to pH. So from this microtiter plate, normally we go further to deep well plates that are around 1 milliliter. Then we go to, we call them baby bottles. These are 200 mLs. And further product is taken to the pilot plant where manufacturing volumes are higher, 3 to 6 and 10 to 150 liters. These steps are important to transfer the product to the industrial scale or transfer this fermentation process to industrial scale, because very often when we do upscaling, the fermentation parameters and the fermentation speed and the result, it actually changes a lot. So it is important to take as many steps in between microtiter plate and industrial scale as possible to ensure that process is smooth throughout the whole way. And in the end, when the product is at the pilot or in the final industrial production, we compare first a big plot where there are a lot of attributes, and of course all of them are important. However, most of the time there are more important parameters that we need to consider, and they are usually plotted separately and compared for different starter combinations. This is done through organoleptic assessment through the trained panel. However, at the moment, in our company and in some other places, people are developing automatic methods on how to do this assessment through key chemicals that are attributes for acidic or grainy or umami or some other notes. And this is done through HPLC and the gas chromatography measurements to increase the throughput of the assessment. On top of that, at these days, what we also do, we use artificial intelligence to utilize all the data that we have collected through the years and years of running these experiments, and we are getting to the point that we probably will not need to run as many test trials in the future, and the artificial intelligence platform will predict which combinations are best for which basis. And that was it on my side.
Thank you very much, Valerie, for your very nice presentation. I understand from your presentation that there is quite a challenge to actually upscale from fermented from microtiter to the food factory. Quite some consequences, I can imagine. Thanks again. I very much would like to invite the other speakers to turn on their microphones and their cameras, because we do have some questions. Amongst others, I just go on row through the questions that have been posed, and I see that for Luc de Vuist, some artisanal or traditional bakery products for sourdough from ancient wheat varieties of their land ranges. Do you recommend this activity or any comment about it, knowing that usually they are characterized with poor technological properties? Probably this goes also back to maybe a question to Christophe about, okay, how does the consumer part in health firm works, and what type of analysis have we already done? Luc, can you provide an answer?
You asked me?
Yeah, I asked you first.
I thought you said Christophe. But can you repeat what exactly you asked?
Now, this is about artisanal and traditional bakery sourdough products.
Yes.
And they are usually poorly characterized technologically.
Yes.
Do you recommend using landrace wheat varieties or rye varieties in sourdoughs, and what are the potential consequences of that?
Okay, but first of all, each sourdough is different, but it is the way you maintain it, you refresh it all that time, which will determine if it is a stable one or not. And there are many bakers which refresh their sourdoughs. There is one in Belgium up to 100 years from generation to generation. Of course, they can sometimes modifications can take place because so many factors influence that, like refreshment time, storage at low temperature or not, repetitive refreshments twice a day, once a week, once a month. It all determines which microbial consortia you will have. And then, of course, there is the raw materials and the ingredients. Some add fruit, some add salt, some add spices, some add even alcohol. It all has different recipes. All that will also determine your consortium. But once you have a stable and major sourdough and you refresh it on a regular basis under stable conditions, it should not change over time. And of course, it is recommended to use that sourdough repetitively. Old land races, that's another thing that will maybe have an influence on the microbial composition as well. I don't know, but I can tell you this month we start a new project on the use of old land races to produce sourdoughs and sour beers as well, to see if that could be of influence, if the flavor of ancient times can come back in our bread and beer.
Wow, that would be a great project. In the past, everything was better, I think. Yes. Christophe, can you maybe go a little bit more in detail on the consumer part, where you actually gathered quite a lot of sourdough samples?
Oh, are you talking about the citizen science project, Jan, or are you talking about consumer behavior and consumer science? Because those are two different things in the project.
Yeah, the citizen science, where you actually ask the people to present their sourdough collections.
Okay, so in the Health Firm project, we had this rather large-scale citizen science project, as we call it, and there we really wanted to tap into microbiomes or microbial consortia that are available at people's homes, but also at small bakers, for example, or larger companies, all those that wanted to contribute. So initially we called it community science. And so we have developed the project for, let's say, over a year, and then started inviting people to send their sourdoughs together with questionnaires on how they use the sourdoughs or fermented foods, how they prepare them, how they keep them alive for 100 years or longer, the age. And so these samples were all sent to different hub labs that are specialized in the microbiology. Luke's lab is one of those. The main lab or coordinating lab was at ETH Zurich, but also University of Helsinki was part of that. So and there, those sourdoughs are really, let's say, taken apart, looking at the individual microorganisms, phenotyping those microorganisms, but also looking into genetics to see what these different microorganisms do, whether we find new ones, and what they can do for us in going into designed microbial communities for health promoting products. So that is in essence what we are trying to do. And I think the stage of getting sourdoughs in has been quite successful. We have 680 sourdoughs. We have over 200 and more other fermented products. So that is what we're doing in the citizen science project.
Mm-hmm. So there is quite an input in the citizen sciences project for sourdoughs. And what is the diversity that you are you able to say something about that? Because that was something where Luc and Kati were actually pinpointing on. So the consortia that you use for sourdough culture have consequences for the products. Do you have can you tell a little bit about what you have noticed at this moment already?
I cannot divulge too much, but what is clear is that the usual suspects, of course, come back all the time. Yeah, I think that's clear. But there's also some very interesting novel strains that are being identified and which we will further look into, go to whole genome sequencing and others. So.
Okay. Kati, there was a question about metabolites. Were any metabolites of nutritional components measured in the bread constituents with fermented wheat bran compared to normal wheat bran? So what is actually the consequence of the pre-fermentation of the bran that you use for the different types of bread that you showed us?
In the context of fermented wheat bran?
Yes.
Yeah, that was actually studied quite intensively because it was part of a big previous EU project, health grain project, and these fermented wheat brans in breads were studied in vivo trials, and there are a lot of ferulic acid, for example, released, and what was shown in that project was that when you consume such fermented wheat bran, that you have more ferulic acid absorbed into your system, and it's more bioavailable in a way.
So in principle, the pre-fermentation, I call it then pre-fermentation, is actually increasing the bioavailability of the components that are usually in the bran.
Yes, and also what it does is that it degrades phytic acid, and minerals are most likely more absorbable because phytic acid is binding the minerals, and then it's degraded in sourdough fermentation when they are likely to be more bioavailable.
So that's very much linking with what Luc was presenting with reference to with reference to the fermentation processes that the potentially nutritional benefits that we get. Valerie, in your experience, are the results from the microtiter plates consistent with the results from upscaled fermentation trials? With regard, for instance, to acidification ratio. So do you see the same if you scale up?
We never see the same, right? So from one base to another, the similarity is either less or more, right? But the more experienced we're getting in the base preparation, the more we have hands-on understanding on upscaling process itself, the more predictable we can get and the less trials we need to smoothly transition from microtiter plates to pilot plant and further to the industrial scale.
Mm-hmm.
Because the change will occur inevitably because of simply transition from pilot to industrial scale. The mixing units, the equipment that is used to pump products around, the homogenizers, and you name it, all the list of parameters that are different. They all have impacts, some smaller, other greater, on the fermentation. However, some of these things that are also possible to model in the microtiter plates. That's why downscaling is so important for fermentation predictions.
Okay, so in principle, you can based on downscaling, you can predict what happens in in the large factory by setting it into very, very small models.
Yes.
So also, for instance, inhomogeneity of your dough or your fluid.
That's right, because if we can model the system in a smaller scale, following the fermentation, then we could also predict how the situation is going to be on the larger scale. And here we're not talking only about adding optical tools how to track the pH. We could also use small electrodes on the robotic systems to measure pH in those systems where we see, for example, a lot of flaking, a lot of separation of phases, and all other things.
Mm-hmm. Christophe, question to you, I think. What are the project efforts to collaborate with more bakeries to incorporate the novel modifications to startup cultures?
At this moment, there are none, to be very honest, in the sense that the EU project, as we have it, it's a research and innovation action. It's a closed project. So that means that we cannot take on new partners, or that we cannot just disseminate results as we're making them. We have to take in account sensitivity of results to some extent. So what we try to do is to be as open as possible and as close as necessary, as the EU wants us to be. But I'm sure that if we find novel microorganisms and novel strains, that through the collaborators and others, they will make it to the market at some point.
Okay. With that having said, I would like to thank all the presenters, Luc de Vuist, Kati Katina, Valérie Gutsaal, and Christophe Courtin, for presenting the very nice presentations on microbiology and technology of sourdough production. There are still some questions open to be answered. We will come back on that, and we will do that by email. Thank you very much for your contribution, and hope to see you once again when we organize a next webinar. Thank you.
Thank you, Jan.
Bye.
Thank you.